Hi guys, I'm Megna,
I'm Aisha. We're in
and
your
we're in your bio, and our project is frequency in bean plants.
and we did an experiment in case you're wondering. Okay, so our first learning goal was how does frequency affect the germination rate of bean plants?
And then our second one was why do plants germinate quicker at their optimal frequencies? It's like the science behind it.
And then just a general overview of our experiment was um, we were just testing on how bean plants would germinate under like certain frequencies. So we tested 3 different frequencies and we wanted to compare it to whole bean seeds that were not growing under any frequency.
Okay, then the personal reasons. So we wanted to see like the effect of something on an organism, and like the most available one was a plant. And then we also wanted to learn about something that we didn't know about because like we know about like water, but we didn't really know about frequency.
Okay, we're gonna get into our experimental design now.
Okay, so basically a lot of this, it's like easier to see in the pictures and it'd be kind of like redundant to say it out loud. So if you want to like look at our materials, you can look at them. But we're going to be talking about like the important things. So one of them is the independent variable, and that's like the frequencies that we were changing up. So we did 3 different frequencies. One of them was the 20 to 2000 hertz, one of them was the 9000 to 11,000 hertz, and then the highest one was the 18,200 to the 20,000 hertz, and those kind of correspond with like low, medium, and high. And we'll be like calling them low, medium, high as we go on, because it's a lot easier to say that than like those numbers. And then our fourth group was our control group, which was the no frequency. So basically, like whenever we tested it, we used those 4 types of, and those are our 4 treatments.
And then, so our dependent variable was, we had 2 dependent variables. So the first one was the height of each seed's radical. And the radical is basically the primary stem that comes out of a seed when it germinates. And then our second dependent variable was the percentage of seeds that germinated in each group. So these were the 2 just variables, data that we collected. Now for the fun part, which is the pictures. Okay, so to start off, this is where we're going to get more in depth with our procedure. So basically what we did is we wet some paper towels and put them on these paper plates, and then each group had 8 seeds, so we spaced the 8 seeds out on these wet paper towels.
Okay, so as Aisha said, we had our 4 paper plates, our beans, our paper towels, and we put each of these like napkins underneath a cardboard box, and we had 4 for like our 3 frequencies and our control. So we had 4 of these cardboard boxes, and then you can scroll down a little bit. Like obviously we had labeled them so we could see them, and then we had covered each box with a blanket, so you can scroll up a little bit if they want to see that. And yeah, we had our blanket because that could like insulate a little bit and keep the sound inside. So that's the setup.
And then, as Magna mentioned, there was obviously each group was put into each cardboard box. So then we had 3 devices because there was 3 treatment groups in the control. So in each treatment group, a device was put in, and through this device, we played the labeled frequency. And on the right are Megna's devices, and on the left are my devices. And so we wet the paper towels also after 8 p.m. every day. So we played the frequencies from 4 to 8 p.m. and then we wet them after 8 p.m. because the napkins needed to stay wet in order for the seeds to germinate. And then also after the 4 days, that's when we collected all of our data. So we measured the radical height of each seed, like I mentioned before, in the dependent variable, and then also the proportion of seeds that did germinate in each group.
Okay, I think the coolest part was our results. So that was our before. We kind of reused the same picture, but okay. And then now this is like, so we each set it up at our own houses. This is mine, and we circled all the ones that did not germinate after 4 days. So as you can see, like in my control, I had 3 of them, and then in my low frequency, I only had 1 of them. But in my higher frequencies, like literally all of them sprouted, so that's pretty cool.
And then because ran the experiment once at Magna's house and then once at my house. And so for me, 4 of the seeds in my control group didn't sprout. And then also, like, I want you to pay attention to the radical height of the seeds, because if you look, it's like kind of cool because the ones in the control group are like a lot shorter than the other 3 groups. And then for the low frequency group, 2 of them did not sprout. For the medium frequency and the high frequency, both of them, 1 of them did not sprout. And again, like, you can kind of see the radical heights are like a lot longer in the higher as the frequency increases. So now we're gonna get into the actual evidence that we collected.
Okay, and then if you want to see those pictures in depth, you could like look at the URL and go to the website if you want. And these are our graphs. So this is the effect of frequency on like the percent of bean seeds that actually germinated. So this like shows you how like the percent of them like that actually germinated. So as you can see in the no sound, it's like a lot lower, like the 56.25% compared to the higher frequencies.
Okay, and then the box plot, we both made a box plot that shows basically it's just the radical height was the other dependent variable that we collected. And as you can see, as the frequency groups were increasing, the median radical height was also increasing. It's kind of close between the frequency groups, but it was increasing. Okay, and now for a conclusion, which is the cool part. So basically, since we didn't experiment, we're obviously going to have some error because we didn't run ours in a like perfect ideal environment or in a lab. So the biggest error, I would say, in my opinion, I don't know if it's Megan's opinion, but is that we used different devices. Not only did Megna and I use different devices, but we also within our own little experiments, we use different devices. So we used 2 laptops and a speaker. And obviously this probably had some kind of an effect on our results. And ideally we would have 3 of the same laptops, but we obviously cannot afford that. So unfortunately we had to use different devices.
Okay, then another one was the noise disturbances. So we tried to keep like our families out of the garages, but that didn't really work out. Like on the first day, my brother thought one of the boxes was like a couch because it had a blanket over it. So, and he sat on it. So that probably didn't help. That was probably a source of error. So that kind of sucked, but it's okay. And then also, like the blankets, we did use them for soundproofing, but like Aisha said, we weren't like rich enough to buy actual like soundproofing box material. So that could have been a source of error since the sound could have like gone other places.
Okay, so now we're going to get into our learning targets actually. So the first learning target, which was how does frequency affect bean germination, we collected 2 different kinds of data to test this. And so the bar graph, which was the first kind, and it showed the percentage of seeds that did germinate. And I think Magna kind of already talked about the trend, but we also ran significance tests to see if the data actually proved that the percentage of seeds that germinated was different between the different groups. And what we found was that there was a significant difference between the percentage of seeds that germinated in the 2 highest frequency groups and the control group. But there was not a significant difference between the percent of seeds that germinated in the low frequency group and the control group. So we kind of grouped it together, like the 2 highest frequency groups had an effect on the percentage of seeds that germinated, and the 2 lowest frequency groups had a lower percentage of seeds or the control group and the low frequency group had a lower percentage of seeds that germinated. And then we get into the box plot, which is it showed the radical height of each group, the radical height of the seeds. And again, we ran significance tests to see if the data was actually proving anything. And we found a similar trend as we did in the percentage of seeds that germinated, and that was that the average radical height of the bean seeds in the highest frequency group was greater than that in the low frequency group and the control group, but the average radical height of the seeds in the high frequency group was not significantly different than the average radical height of the medium frequency group. So again, we kind of grouped it was like the medium and the high frequency had a higher average radical height of the seeds than the low frequency group and the control group. And then we're going to plug some other studies here because science is cool and you should read other people's papers when you do experiments. So the main reason we did bean plants is because, one, they germinate quick, and two, because we didn't find any other experiments that specifically tested on pole bean seeds. And the interesting thing is, if you read some papers about this topic, is that each kind of plant has their own optimal frequency that they grow the quickest at. And for example, tobacco plants grow, their optimal frequency is 400 hertz. So what we found in our experiment was that higher frequencies are more optimal for pole bean seeds, which may not be the case for other plants, but it is for pole beans.
Okay, so this is our second moving target, and that's more like the science behind what's happening. So the most important thing to keep in mind is that like you and I have like ears, but plants do not, because nobody really knows like how plants hear. There's no like identified mechanism. So a lot of these theories are just based off like observation of what's happening, but nothing can be like fully like 100% confirmed. So one of these is the stomata theory, and basically like we know what stomata is. Like they do the gas exchange, they let water come in, they can also absorb some like nutrients. So basically, one theory is that like at a certain like optimal frequency or higher frequencies, there's going to be a gene that gets activated, and that can code for a protein that expresses this activity that can widen the stomata even more than it is. And a wider stomata can mean like more water is coming in, more nutrients, and that can be really good for like metabolic function, which could explain like why they're growing more under optimal frequencies. Another one is the membrane fluidity theory. So according to this one, at like a certain optimal frequency, the secondary structures of the proteins are going to change a little bit, and because they're changing, this alters the fluidity of the membrane. And if a membrane's more fluid, this can mean that there's like molecules coming in, more lipids, more proteins, and in turn that makes the plant like healthier, which can also explain why it's growing bigger. And then another one is the superoxide dismute. I'm just gonna call them SODs. And basically, whenever something's like exposed to oxygen, there's a chance that it can become oxidized in like a bad way, which can harm a lot of things. And SODs are actually enzymes that can stop all this like bad stuff happening because they are antioxidants. And at certain optimal frequencies, it has been shown that more SODs are able to be produced. So that could be like helping the protection of the plants.
Okay, and then the last theory is the ATP theory, which basically is that when when there's a higher frequency, there's more ATP production in the plants. And obviously, more ATP production leads to more growth because if there's more ATP, then the seeds have more energy to expend on actually growing and having longer radicals. Okay, and then lastly, we're going to talk about the application and purpose of our experiment, which is like, why does this experiment matter, aside from the fact that science is cool and we always want to discover new things. But practically, this experiment is very helpful in the real world because bean plants are grown. They're like a very important crop and they're grown widely in the US. So approximately 1.8 to 2 million acres of beans are grown in the US. So you can imagine how many bean plants are grown. And this study can be used and utilized and implemented when farmers are commercially growing bean plants because they can speed up the germination process of these bean seeds. And this saves both time and money in the long run because obviously the seeds are germinating faster, which means that you can actually germinate more bean seeds in a specific amount of time, which means that you can sell more bean seeds and make more profit.
Okay, and then some more like indirect applications are a pretty cool thing is that like we were talking about how frequency affects plants, but there's also effects of frequency on animals. And the biggest one is that like at frequencies, at like different frequencies, animals are actually attracted and want to go towards that sound. So this could be helpful because it has been shown that when you do play like some types of frequencies, birds and butterflies are gonna come and they're gonna help get rid of like the pests that are near the plants. And that could be helpful because, you know, like less pests means that more plants will actually grow. And that could help out the farmers who maybe, you know, don't have to worry about like pesticides and these things that are getting kind of controversial. And then another one is hummingbirds. So basically, like hummingbirds are pollinators, and hummingbirds are like attracted towards frequencies. It has been shown. So with like more hummingbirds coming in, there's more pollination, which can mean like more plants are being produced. So that can help out a farmer. And then also, it has been shown that with more pollination, you can have like bigger fruits, and that would also be pretty cool for a farmer who's selling bigger fruits. And yeah, I think that's basically our presentation.
Yeah, if you want to see this website, the link is up here. It might also be in the description if this is a YouTube video. If it's not, well, you can ask us for it. We spend a lot of time on it and we really like it, so
Yeah, it's pretty cool actually.
yeah. Yeah, and if you stayed till the end, you can text Megna and she will
I
give you.
got this ready. You text me. Whoa, you guys have the coolest website, and you get entered in a giveaway. And then if I pick your raffle ticket, you get a $5 T4 gift card.
Sounds awesome, right?
Awesome.
Okay, yeah, so if you want to check this website out, you should. If you want our paper, you could ask us, we'll share it with you. But we hope you enjoyed.
Bye.
Okay, you stop recording.
Oh, okay.